In this chapter, we're gonna learn about the two subjects, folding and structural dynamics. Protein folding uh, could yield uh, native three-dimensional structures of proteins, starting from the unfolded polypeptide made by ribosome based on the messenger RNA. The active form is actually correctly folded native forms, not the nascently made unfolded proteins. So this protein folding process is called sometimes second translation. It has been known that folding information is encoded in primary structure of protein. The second topic is dynamics of proteins. A protein in its native state is not static. They just undergo small fluctuation to the large scale of changes. First, we're gonna look at protein folding issue. In genometer, given that we do have so many protein sequences, prediction of 3D structures and its related property from its residue sequence would be very powerful tool in the protein area. When unfolded proteins fold into native form, actually both enthalpy, indicating bond formation, and entropy increase during folding. Basically, this process is spontaneous reaction, but sometimes requiring the helpers. Protein stability is called marginal because the Gibbs free energy changes only -5 to -10 or -15 kcal per mole between unfolded and native form. That's going to be very little. Note that the hydrogen bond single is only -2 to the -5 kcal per mole. Then why proteins is so marginally stable? There are certainly logical reasons. Cells must be able to synthesize them easily, and cells must be able to alternatively degrade when they are not required. And also many the functions of protein require structural flexibility. Protein folding initially thought to be achieved by random searching model, meaning proteins just experience all possible conformations and reaching most stable conformations. Here, the simple calculation about the 150 raised to long protein, how many possible conformations, assuming that a single residue may have 3 different conformations giving this kind of number, and times 10 to the minus 12 the magnitude, that is time required to change it from one structure to another conformation, will give this number. Okay, compare with the universe age of this. This is too big number. Okay, meaning any single proteins cannot experience such all possible conformations to reach native forms, because mostly all proteins just reach native forms within 0.1 or 100 seconds for the slowest, the special case. So this is called Levinthal paradox. So another choice is called kinetic model, which tells what protein folding just occur through some the kinetic pathway of limited number of some intermediate. Okay, that's more logical. It has been known that during folding process, a third species called molten globule (Mg) appears over and over through different proteins. What is the feature of Mg? It has significant amount of secondary structures like alpha and beta, but not real compact three-dimensional structures. In other words, loosely packed hydrophobic interaction. In general, during folding process, initial the interactions by hydrophobic residues tend to be buried in the interior. That's going to be greatly restrict the possible conformations. Proteins undergo a single pathway for protein folding or multiple pathway, as shown here. As an example of single pathway, as in the banase, which is a small bacterial ribonuclease, basically has 3 alpha helices and 5 beta pretty sheet in C terminal. It shows certain the intermediate that may be characterized by it may have a secondary structure and some location of alpha and beta, relative arrangement of beta. For the multiple pathway example, lysozyme, which has 2 domains, alpha, and the other domain is 2 alpha in red and 3 betas with blue colors. Okay, some of them are different in the middle, intermediate, but the recent later development just lead to the more comprehensive view called ensemble model. This is just two-dimensional picture for this one. Unfolded pole actually exists in so many different conformations. Each individual undergo a different route to reach to the native forms. Okay, this is just the energy the diagram from starting the unfolded and molten global and the native forms, but it could reach to the some transition higher energy barriers. The three-dimensional picture called funnel model in this category to visualize more the features. On top, smooth lines represent again the unfolded forms with different conformation. They just follow the steep, nearest steep. So Y scale indicates the free energy level, and this scale indicate the number of conformations, and they just go and then meet some energy barrier depending on the track. Some might be trapped, but some might just undergo very easily and reach to the certain native forms. In this area, there is some competition had been held called Chapt, Critical Assessment of Structural Prediction, starting in 1994, and then every 2 years just opened. So the recent one will be 10th, held in 2012. Basically, they just give primary sequences of certain protein that had not been known at all for their three-dimensional structures. But the organization already knew the answer for the native form, and then within certain period, the people just let them just predict theoretically, not by experiment approaches, and then they just try to promote people in this field to compete. Okay, so this is one example obtained by David Baker in University of Washington in 2006. The colored obtained by the experimental approaches, and the gray colors, the one is obtained by theoretical approaches. Okay.